So now we're into the next chapter, and the first topic in the next chapter is diffusion in water and molecular versus advective transport. This is very related to the previous chapter, and this is referred to as eddy diffusion or transport diffusion. And the first point here is fixed law of diffusion. And there's a general law of diffusion, and what it states is the rate, the flux, so that's this flux here, and this, if we have a concentration of the red stuff, right, and the solution is yellow, so as it moves into the red stuff moves into the yellow, it makes it orange, right? So we have this flux from high concentration to low concentration. The material in high concentration has more potential energy if there's an area near it that has low concentration. The two things that influences this is how much of a difference in concentration there is, number one, and number two, how far apart these are. So the further apart these are, the slower the diffusion, because it takes longer to get from one place to another. So we can think about this in terms of an analogy. I use the analogy of a pool table. If you have a really regular sized pool table, right, and you take the pool balls and you put them in one side of that pool table, and then you get a small sledgehammer or two, and you start pounding on the edges of the pool table. Not enough to break the table, just enough to shake the living bejeebers out of it, right? What happens to the pool balls?
They'll start knocking into each other and kind of separating.
Right, and what's in the end, if it's a completely flat pool table and if you've blocked off all of the pockets, what's going to be the distribution of the balls?
Just even in distribution?
Right, so at the beginning you'll have a diffusive flux. You'll have pool balls moving from into the area of low concentration from the area of high concentration. Right? Okay, so there's the first part of the analogy. Now let's say I have a pool table that's 5 times the normal size, but I have the same number of pool balls. And I can put the same amount of shaking energy onto that table. What's going to happen? How quickly are you going to attain the same state relative to the smaller pool table? That is, you had an even distribution at the end, eventually even distribution. Will that happen more quickly or less quickly than it did on the smaller pool table? Got some answers here. Less quickly, right? It's going to take, you'll be there all afternoon. Right, it's going to take more quickly. Okay, the one more thing I'm going to do with this is I'm going to say I've got the pool table. I'm back on the small pool table now, and I was doing it like this, right? I was pounding on that pool table. Now I'm pounding like this, like 3-4 times as fast. What's going to happen to the flux rate? How long is it going to take that to go? Will I be there all afternoon? More quickly, right? So what am I actually doing when I'm hitting it more quickly, when I'm talking about diffusion of molecules with the faster pounding? What am I trying to get? What analogy am I trying to get at there? Maybe more flow, but probably not.
Is it like reaction rate?
Sort of. I mean, reactions happen more quickly because of the same feature. More vibrations, more knocking, more diffusion, right? Not more potential energy in the sense because the concentration difference is still the same. What else do we got going here?
More turbulence.
Sort of. Yeah, sort of more turbulence. But let's say we're a really small scale and there's really not much turbulence at all. But we've got really small Reynolds numbers. Oh, let me ask the question differently. Let's say you're making iced tea. When do you add the sugar? You can either, you heat the water, you put the tea in, and then when do you add the sugar if you want to sweeten it? Okay, so Abigail's going to add her after it's brewed. Cassie says while it's hot. So you add it when it's hot or cold?
While it's hot.
Yeah, right. So you add it while it's hot. Why do you add it while it's hot?
I
Easier
mean,
to,
because it dissolve more sugar inside of a hot substance.
Right, and it'll dissolve more quickly. And why is that? What's happening with the molecules?
Higher interaction rates.
Yeah, they're moving faster. So to get back to what Lane said, this is the same reason we've got the Arrhenius effect, right? You actually have all the molecules are moving faster and chemical reactions will go faster as you increase temperature. So what I'm trying to do is getting more temperature higher temperatures will increase rates of diffusive flux, even in the absence of turbulence.
So can you just explain what, like, exactly what the C and the X are when you look at the equation?
Sure. So we have some spatially separated, two points that are spatially separated. Concentration 1 is the concentration of higher concentration. Concentration 2 is a lower concentration. The distance, x1 minus x2, is the distance between those 2 points, right? So the change in concentration divided by the change of distance times this constant will give us the diffusive flux. Does that make sense?
Yeah, that makes sense.
Yeah, so if they're further apart, less flux. If this concentration is higher than this concentration, more flux. So what does that mean? Like, you know, as far as bacteria that are living together, are they going to have a high flux from one to the other or a low flux? High diffusive flux or a low flux, diffusive flux? Low, right, Emma? Because they're so close together. They're microns together, microns apart from each other. So much microbial communication and interaction happens via chemistry. Now that's in the absence of turbulent flow, and turbulence can move things along pretty well. And we'll talk about that, I think, now. Okay, so molecular diffusion is way slower than we necessarily think it is in our brains. And this is going to be the demonstration that we have for the laboratory. And this is agar plates. And the agar is a polysaccharide from seaweed. And you can make a solution of about 1%. And it'll be, it's kind of like gelatin. It'll make this solid stuff. But really what it is, it's a polymer that goes all through the liquid. And even though there's only 1% of the actual compound in there, right? So it's very low. It's keeping turbulent diffusion from flux from happening. Stated differently, the space between those polymer strands is so small that Reynolds number is small. So what's high?
viscosity.
So viscosity is high, right? So you're just not going to get much mixing. So this is basically at our scales, and you know, and on the screen they're a little bit bigger than, but not much bigger. At our scales, we're basically saying, okay, this is what it's like in a very viscous world. And I put a little dot of dye in the center of this and let it go for an hour. See, you can see it kind of cut out a hole there. A day later, it didn't go out that far. By 2 days, it's getting to the edge, but it takes 2 days. So if you had that glass of tea and you put in a sugar and you could keep the mixing from happening, because what's really going to happen if you have water, it's evaporating off the top, and then there's these currents that are occurring. So there is some turbulent mixing. But if you could stop that from happening, it would take days and days for that sugar to get into that solution, just because molecular diffusion is happening at such a slow rate. This is going to be important when we talk about stratification of lakes, what's happening in sediments and diffusive flux in many places. So now we can take that concept of the flow boundary layer and expand it to the diffusion boundary layer. But the diffusion boundary layer is much smaller. Turbulent flux is thousands of times faster than diffusive flux. So if you have turbulence, it's going to completely override any diffusion, molecular diffusive flux, right? It's going to move things, that is turbulent diffusion, if you want to think of it that way. It's going to move things so much faster. That's why you stir your tea. You want to increase the turbulence and the movement of the of the water, or your coffee, or whatever else you're trying to dissolve sugar in. If you don't stir it, you don't break down that flow boundary layer, right? You do not, you have molecular diffusion trying to get the sugar to dissolve out of the particle, off the particle and into the solution. If you stir
So
it,
does
go
so
ahead.
does turbulence essentially have the same exact molecular effect as heat, like just batting everything around?
It has even more of an effect. Yeah. So it's you know, heat, you can double, triple, quadruple, but not thousands of times higher rate. So it'll override. It's even more important than heat as far as mixing of materials or movement. So basically it's similar to the flow boundary layer, but defined by the region where molecular diffusion dominates. And if you can think about that no slip zone, right, that I talked about, that is going to be molecular diffusion in there. But once you get up into the laminar flow, that's going to be moving things along much more quickly than any of the molecular diffusion can. So this relates to biogeochemical constraints as well as physiological constraints. It really controls how organisms interact with the environment, shapes and sizes of organisms, as well as things like how quickly a chemical will diffuse out of a sediment or across the stratified layer of a lake. One of these areas that we can talk about is the surface area to volume ratio. This is really an important aspect of the physics of how organisms evolve and adapt to their environment. So let's consider two cubes, one that's a micron on a side and one that's 1,000 microns along a side. Right, so surface area of a cube, how many sides are there? How many sides does a cube have?
6.
6, good. So if there's 1 micron on each side of a cube, right? So each side of the cube would have 1 micron squared. So there's 6 microns squared of surface area on a cube. The volume of the cube is 1 times 1 times 1 micron is 1 cubic micrometer, right? In contrast, the 1,000 micron cube, which is also known as what unit? 1 what? So microns 10 to the minus 6, right? So 1,000 of those is 10 to the minus 3rd meters. So that is not a meter.
Millimeter.
A millimeter, good, got it. Okay, so we've got a micron and a millimeter cube. The millimeter or 1,000 micron cube is 6 million cubic microns, right? So their volumes are 1 in 1 billion cubic microns. So the surface area to volume of the small cube is 6. The ratio of those two is 6 versus the surface area to volume ratio of the large cube is 0.006. So which one can interact more quickly diffusing into the environment or taking nutrients out of the environment or oxygen out of the environment? Which one can interact more quickly with the environment via diffusion? Emma's guessing the bigger one. Does anybody want to convince her otherwise? Tom, why do you say that?
I'm just thinking back to like regular biology where when you have a smaller surface area to volume, it's easier for you to move around and transfer things in and out with that ratio, basically.
That's sort of it. Abigail said something that doesn't have to do with surface area again. Yeah, definitely does. So let's say we're talking about moving a chemical across a cell membrane. What moves chemicals across cell membrane? I don't know if you want to take ammonium or something and take it from outside, a charged particle from outside that you need as a nutrient to inside. What does it take to do that? What molecules are used to move materials across cell membranes?
Proteins.
Proteins, right? And it may be a protein that takes energy. So you can only cram so many ammonium uptake proteins onto a cell membrane. So if you have a very large surface area relative to your volume, you are able to bring in quite a bit more ammonium relative to your volume. In addition, once you get it in, another way of thinking about that is the average volume, average spot inside of the small object is much closer to the surface, right, to every surface than the stuff in the large object. So, Emma, I'm sorry to say that Tom was correct in that one. Right, so small cells are much better at moving large amounts of materials out. Please don't leave, Emma. Everybody has a chance to be wrong in this class. I will certainly be wrong in this class as well. All right. Yeah, so this is one of the reasons that multicellular organisms are up against a big problem, right? Remember how slowly I said molecular diffusion happened. I had those molecules in that Petri plate, which is much smaller than us, right? Taking days to get from one spot to another. Yet we have to respire. Every single one of our cells has to respire, right? Every single cell in a fish has to respire. How do they get over that limitation of molecular diffusion, getting oxygen into every cell in their body? How does that fish get over that fact that we have really small surface area to volume ratio?